SRM Dental College, Ramapuram, Department of Microbiology presents a video on hanging drop method. So, hanging drop preparation. Hanging drop preparation, the aim of the study is one of the common technique used in microbiological laboratory. It is used for rapid assessment of the motility of bacteria. Careful observance also gives clues regarding morphology of the organism. We can also infer the type of motility which is characteristics for particular bacteria. Procedure, it requires a slide which is known as concavity slide, glass slide with a shallow circular concavity in its center, then a cover slip, a loop, inoculation loop, suspension of bacterial culture, Vaseline and light microscopes are the requirements. So we can see the picture of the cover slip, then bacterial culture, wire loop for inoculation into the cover slip, and also the microscope. Stepwise procedure. Step 1, place a loopful of the culture to be tested in the center of the cover slip. Then hold a clean cover slip by its edges and carefully dab Vaseline or wax or paraffin gel on the corners of by using a toothpick. If too much Vaseline is used, skews towards the center and mixed with the water drops. So next one is turn the clean concavity slide upside down over the drop on the cover slip so that the Vaseline seals the cover slip on the slide around the concavity. Turn the slides over so the cover slip is on top and the drop can be observed hanging from the cover slip over the concavity. Then place the preparation in the microscopic slide holder and align it using the naked eye so an edge of the drop is under the low power objective. So this is the picture showing the concavity slide, the placing the microbial culture in the cover slip. Then you can see the cover slip in which petroleum jelly or the wax is applied. Then by using an inoculation wire loop, microbial culture is placed in the center. Then the slide is placed above the concavity slide and it is turned over. So invert the concave depression in the glass slide to produce hanging drop of the specimen. So that water droplets can be seen hanging. Then next, turn the objective to its lowest position using the coarse adjustment and close the diaphragm. Look through the eyepiece and raise the objective slowly using the coarse adjustment knob until the edge of the drop is observed as an irregular line crossing the field. Step 8, move the slide to make that line passes through the center of the field. Next, without raising or lowering the tube, swing the high dry objective into position. Then observe the slide through the eyepiece and adjust the fine adjustment until the edge of the drop can be seen as thick dark line. This is the picture of the water droplets, the edge of the water droplet where you can see the bacteria, moving bacteria as well as the non-motile bacteria also. If it is the specimen is non-motile bacteria, you can see the non-motile bacteria or the motile bacteria. This is the light microscopic picture in which the edge of the water droplet is shown. Step 11, focus the edge of the water droplet. Drop carefully and look each side of that line for very small objects of that are bacteria. The cells will look like dark small rods or spheres. Then step 12, adjust the light using diaphragm lever to maximize the visibility of the cells. Observe the cells, noting the morphology and grouping and determine whether true motility can be observed. Brownian movement should be visible on slides of all the organisms, but the motile bacteria would also show true motility. So true motility and passive drifting. True motility move in different directions and change their positions. Passive, all organisms move in the same direction. So Brownian movement is an oscillatory movement about nearly fixed point possessed by all small bodies suspended in fluid and due to irregularities in the bombardment by molecules of water. Then types of motility: darting, tumbling, slow and straightly, sluggish. Example: Vibrio cholerae, Listeria, Bacillus, and for sluggish, Escherichia coli. Then motile bacteria: Escherichia coli, Vibrio cholerae, Salmonella, Pseudomonas, Proteus, Citrobacter, Enterobacter, and Enterococcus cassiflavus. Non-motile bacteria example is Klebsiella, Acinetobacter, Shigella, Staphylococcus, Streptococcus, Neisseria, Mycobacteria, and Corynebacteria. Other methods of detection of motility is mannitol. So it is a semi-solid agar which is used for Salmonella, Craigie tube method. Then unstained wet mount and examination under dark field microscope example for treponemes, and motility denotes the presence of flagella. These are different types of flagella which we can see in a bacteria like Amphitrachus, Lophotrichus, Peritrichus flagellated bacteria can be seen. Conclusion: motility of the bacteria decides the type of organisms and thereby the choice of tests to be followed to identify and confirm the organism for lab diagnosis. Safe disposal: the bacteria in the slide hangings, the slides can be infective, so it should be disinfected for 5 to 10 minutes and then it should be washed. The bacterial culture suspension to be disposed of also should be disinfected. Thank you.